Thanks to everyone who's joining us in real time, and hopefully for those who are joining after the fact, this will be interesting for everybody. My name is Sarah. I am a PGY1 at the Hammer program, and I also have the privilege of being a RAMS board member and liaison to the research committee. So we're super excited to bring you this installment of the Research Learning Series that we hope is especially geared towards trainees who might have an interest in starting to make research, and particularly some of this exciting clinical innovation and research translation, some of their careers. We have two experts in the field who are here to talk to us tonight, and their bios are like pretty beefy, so I'll do my best to summarize them, but certainly please feel free to add anything that I missed. Up first, we have Dr. Chris Cabral. He is a practicing emergency physician at MGH, and he has more than 20 years of experience in performing research, particularly into the epidemiology, diagnosis, and risk stratification and treatment of cardiovascular disease, and specifically venous thromboembolism. If you've heard his name before, it may be in the context of such fan favorites as the PERC criteria or the PERT-PE response team that was piloted by Dr. Cabral at MGH and has since made its way both nationally and internationally. He is still someone who is actively involved in focusing nationally and internationally on studying genetic and population risk factors to VTE, and helped to co-found the International Network for Venous Thromboembolism Research, which focuses on genetic epidemiology of VTE. More recently, his work explores biological mechanisms of thrombosis and integration of high-throughput omics with clinical data and machine learning to facilitate biomarker discovery. And as he'll touch on, he has integrated that into a new fellowship at MGB in clinical innovation and research translation, of which our second speaker tonight, Dr. Drew Berincott, MD, Master of Philosophy, is the inaugural fellow. So Dr. Berincott is not only a practicing emergency physician, but he's also a biomedical engineer who holds a doctorate in engineering science from Oxford University, has used signal processing and AI to estimate respiratory rates from cardiac waveforms. His doctoral work has also received international patents and has been licensed by startup companies for use in multiple medical devices. He completed medical school at Stanford University and residency at Hammer as well, and is now a member of the MGH Hospital Center for Vascular Emergencies and the inaugural fellow for the Clinical Innovations and Research Translations in Vascular Emergencies at MGB. So thank you so much for joining us, and I'll hand it off to you guys.
Wow, that was beefy, and I apologize for making you say all that.
Thank you so much, Sarah.
All right, so with that wonderful introduction, I'm going to turn it right over to Drew. He's going to kind of tell you what we're going to cover.
Yeah, so thanks everyone for being here. Our learning objectives for the talk today are sort of several fold. First, we want to introduce what precision acute diagnostics is, kind of a nebulous term that gets thrown around. So we want to introduce that term and sort of provide some definition for it. We want to evaluate the pearls and pitfalls of large data collection, and that's sort of another buzzword that is being bandied about right now, but we want to talk a bit more about what that is and how to do it well and best practices. We want to describe appropriate and inappropriate applications for modeling with artificial intelligence. We want to explain the basics of high-throughput omics and the type of omics, and we want to discuss how to build an EM career in clinical innovation and research translation. For our first topic, we'll discuss precision acute diagnostics, and I'll hand it back to Chris for that.
Yeah, so I mean, this is a term that probably most people haven't thought about. There was a point where I thought maybe we made it up. And so what are we really talking about with precision acute diagnostics, and why is it important to us as emergency physicians, right? The obvious answer to why is it important is that diagnostics is primarily what we do, right? We do resuscitations and we run traumas, we do procedures and so forth. But on every patient that comes to the ED, we make a diagnosis. And some of those diagnoses are easy and some of them are difficult. You know, when I spend most of my time thinking about pulmonary embolism and deep vein thrombosis, this becomes very apparent because we know that, yeah, we treat PE, but only about 5% of the workups we do for PE are actually positive and require treatment, whereas another 5% of all ED patients are worked up for PE. So, you know, there are about 130 million ED patients seen every year, and fully 5% of them, or about 7 million patients, are worked up every year for a PE, and very few of them actually have the disease. So it is critically important, and I think more important for us to understand how to properly diagnose things than it is to necessarily treat them. Obviously, treatment is important. I'm not downplaying that, but we are diagnosticians, right? And historically, where do we get the tools to make those diagnoses, right? We get them from the lab and we get them from our radiology colleagues. And I think now we have the ability and the tools and the maturity as a specialty to start thinking about developing those tools ourselves and owning the space that we should own as clinicians, as master diagnosticians. So, what are precision acute diagnostics? And this is kind of our own definition. It's the combination of big data sources and advanced analytic methods like machine learning and AI to create and validate novel diagnostic tests for patients who present with acute undifferentiated illness. In other words, the typical ED patient, right? And so, what are the big data sources you're going to use in your precision acute diagnostics? Well, obviously, clinical data, right? That's available in our electronic medical records. And we're going to use biomarkers. And those biomarkers might be things that you're used to thinking of, like lab tests. They may be things that you may not think of as a biomarker, like a radiologic marker, right? Or a radiologic finding. But we also have some new tools that we're going to talk about later in terms of high throughput omics that will allow us to screen a large number of patients and a large number of potential biomarkers all at once to identify novel biomarkers and biomarker panels. So, how do we think about this, right? So, the first data source that I mentioned is the clinical data. And so, imagine that you have a large clinical database of patients who are tested for possible PE, right? And now you apply machine learning to that database, and you can actually, because your data set is so large, calculate a pretest probability of PE like we do with the Wells score or any of the other scores or your Gestalt, but like we do now, except now instead of categorizing people as high or low or a Wells score of 4 or 2.5, we can give you a precise exact estimate of what the patient's pretest probability of PE. Now it's 47.3%. And we can take an existing biomarker that we have and apply, say, like an age adjusted or years adjusted or pegged adjusted or pretest probability adjusted threshold to that D dimer. But now instead of doing that in a very coarse way, like simply doubling the D dimer, we can give every patient their own threshold for what's positive for their D dimer. So if your pretest probability is 47.3%, then maybe your threshold for the D dimer should be 372. Right? We can do that now with the large data that we have available to us. What's really interesting is that we can also then do a similar thing for biomarkers. So imagine that you've collected blood samples from patients tested for possible PE. You can now use high throughput omics, for example, say proteomics, although there are others, and you can analyze 5,000 or more individual biomarkers to see which ones are upregulated, downregulated, and how they're moving relative to each other. And those can be used to not only identify individual biomarkers, but with the tools we have available in machine learning and AI, we can now simultaneously analyze those biomarkers and come up with a biomarker milieu or a panel that says, well, if this one's going up and this one's going down and the next one's staying constant, that means PE. But in a different patient, maybe the first one's going down and the second one's also going down, but the third one's going up a lot. That may also mean PE, or it may mean PE is ruled out. So no longer are we going to have to think about a biomarker as a troponin or a D-dimer. It is going to be this whole picture of a patient's physiology when they come into the ED, and what does that mean? And of course, we're going to be able to combine that clinical data that gives us an assessment of the patient's presentation and their pretest probability and their risk with those multi-omic biomarkers and put that all together into what ultimately becomes precision medicine or precision acute diagnostics in the ED. And that's kind of our vision for how we think our science as emergency physicians should move forward. And so the first step of that is large data collection. And here I'll turn it back to Drew.
All right. So large data collection in healthcare is essentially how do we collect all of this data on our patients to sort of figure out what's going on and to build into some of this precision diagnostics. So there is healthcare data all around us, and there is healthcare data that is readily available at our fingertips. We have healthcare data from government agencies that are collecting large emergency medicine data sets. We've got insurance records. We've got databases that are de-identified specifically for training machine learning models. We've got data from clinical trials, which is smaller but generally higher quality as it contains more information about the patients. And then we have the nebulous one, which is the electronic health records, the softwares that we use every day in our clinical practice and the data that's stored in that. And so if we go to the next slide, we have some examples of all of these things. Data.gov, the NEDS database of emergency medicine, CMS data for healthcare records, Epic and Cerner, and then the Mimic sort of data sets are all fantastic sources of emergency medicine data. Going on to the next slide, what defines big data? And so this is a term that gets thrown around a lot. And you get data and you wonder, is this big data and how does that change? And long story short is if it's big data or not, it doesn't necessarily change, but there are some things that are used to define big data. So first is the volume. It must be a large quantity of data that is continuously produced. So something where more and more data comes out, each and every day. And that's where the second piece of this comes in, velocity. And so more data is produced in real time, and it must be analyzed as it's produced. And so that real time can be over seconds, such as monitor data, or it can be over the course of days as clinical courses are updated in the healthcare record. And then finally is the variety. And so data can come in many different formats, and big data has all of those formats, including structured and semi-structured. And so if you guys have ever filled out a note where it has drop downs, that would be considered semi-structured data, whereas structured data is vital sign data, and then there's completely free text data, and all of that would be included in big data. And you have to figure out how to incorporate all of that as you build, predictive models. So what are the benefits of large data sets? So importantly, it allows us to do precision medicine. And Chris sort of alluded to it already. And to bring back the example of PE, when you think about PE, you think about the pretest probability. Precision medicine allows us to drill down to that pretest probability to an actual number as opposed to a broad bucket. It allows for preventative medicine. If we have a large population of patients that we know develop a certain type of cancer, can we take that population and can we try and figure out who looks like them 20 years earlier and predict that they may have also that cancer or they will develop it and then use that for preventative medicine? And finally, we have the ability to study rare diseases. And so when we think about PE, there are certain risk factors, but when the original models were derived, we didn't have enough data to sort of incorporate those into the models. Whereas if you think about something like factor V Leiden, not a lot of people have that, but if we have enough data, we can actually build that into a model in a way that we have not historically been able to.
Yeah, so just as a personal story, kind of a color commentary, when Jeff Klein and Mark Courtney and I were working on the first version of the PERC rule before it was validated, we combined our data and we recognized that there would be certain entities, certain patients even, who had rare conditions or a combination of rare conditions, like a pregnant person with factor V Leiden and 3 family members that have had DVTs that wouldn't necessarily light up in a regression analysis because they were rare, right? And so that's why, or at least one of the reasons, that we allow clinicians to say, you know what, we're not going to use the PERC rule because this patient is not, in my Gestalt estimation, low pretest probability for PE. That's why that's there, because if you see that patient who's pregnant with factor V and 5 family members that have DVT, you're going to say to yourself, I'm not going to PERC that patient out. Well, we had to build the PERC rule that way to account for the fact that we didn't have the type of patient volume that would allow us to study rare diseases, but with big data sets, we do.
However, on the flip side of that coin, large data sets do present several challenges. And if only they, were just for all benefit, but they're not. And there's a lot of challenges, and they're not challenges that can't be overcome, but they are challenges that must be carefully considered when someone is using large data to build models to develop precision emergency medicine, acute diagnostics. And the first of those is computing capacity. And, I'll mention this sort of historically, honestly, a lot of the things that we're able to do, and the reason that we've seen a real growth in artificial intelligence in the last 10 years is our computing capacity has gotten astronomical. There are probably things that we may think about trying to do in emergency medicine where we don't have the computing capacity. By and large, a lot of the things that we want to do, we do have the computing capacity, which is phenomenal at this point. But a big challenge and a big consideration is incomplete, missing, and contradictory data. Unfortunately, these large data sets have limited human quality control. If you run a research trial, there is probably someone that is sitting down and hand transcribing data, which can present errors in and of itself. But one of the challenges with big data is if it's being pulled from a computer and just pushed into a database, if the computer is erring, it's going to keep pushing that erroneous result into the database over and over again. And so you have to really think about finding those errors. One benefit is oftentimes in these large data sets, there are patterns to those errors that you can find and correct. But a big challenge can be, for example, in payer data with ICD-9 and 10 codes. And I know Chris has another anecdote about this and where the challenges arise.
Yeah, this became evident to me 2 decades before we had AI. One of the earliest studies that we did, we were trying to identify every patient that came to the ED who was diagnosed with a PE. And so we looked at the radiology reports of scans and we looked at VQs because it was a long time ago. And we looked at ICD-9 codes to see every patient that came through the ED who had a coded diagnosis of a PE by the time they left the hospital. And what became very quickly apparent was that about 2/3 of those codes were wrong. Most of them were patients that had a rule out PE as their ICD-9 code. Some of them had been treated for a PE while they were in the hospital, but that PE had been diagnosed on a previous visit, and simply because they were getting their anticoagulant, they were considered to have a PE during that hospitalization. So there are real problems with certain administrative codes that we know have existed for a long time and now can just be amplified by the creation of these large data sets if they don't have appropriate quality control.
Amazing. Another challenge of large data sets is the lack of granularity in some data sets. And this is not all data sets, but this comes down to thinking about the question that you're trying to answer and thinking about the data that you need to answer that question. So, for example, if you are using a claims database, that claims database is probably not going to have some of the discrete details about the emergency medicine or the emergency department visit for that patient that you may need to predict something that occurred in the emergency department. And so you really have to think very carefully about the data that you're using and making sure that it has all the details you need. And sometimes we have to make decisions where you get less patient records, but more granular data. Or in some cases, you want more patient records and less granular data, which is a decision that has to ultimately be made. Sometimes, though, we do try to merge those data sets, which presents its own challenges. If you have a patient record in two data sets, say in a claims database and then in the electronic health record, and you want to merge those, you have to be very careful and know that there is some unique identifier in those data sets that will allow you to merge them. But sometimes that's not possible, and that makes answering some of the questions that we want to answer even harder. And finally, you have to think about data bias and ethical considerations. So people talk about how you can build AI models and how they are biased. They're not. AI models themselves are not biased. They are just taking the data that's given to them and spitting out an outcome. They're just statistical methods. However, it's the data set that can be biased. If the data set is not representative of the population that you're trying to predict something for, it's not the right data set. And sometimes you have to then sort of narrow the model and say we're only trying to predict in the population of Boston, say. Or you have to expand the data that you have and try and get a more representative sample of your data so you can create data sets with less bias. And then the ethical considerations come in, and you want to be very careful because we are using patient data, and often it is de-identified, and hopefully we would never be able to track that back to, the individual person themselves that the data was collected from. But you do want to keep that in mind as you're using all of these things and take special care. So all of that to say, the challenges of large data are a garbage in and garbage out problem. And you may have heard this before, but if any of those challenges arise for the data set that you're trying to use and you can't figure it out and you can't sort it and you can't fix it in some way, essentially any model that you create, you're just putting garbage in, it's going to spit out an outcome and you have garbage out. So you can't take a bad data set and get a good predictive model. It's just never going to happen. So a quick example of analyzing large data sets. And I bring this up because it's sort of a multi-step process. And a lot of times people think that, oh, I'm going to do this, sort of analysis and, it's going to be sort of ready to go. But there are many steps that you have to go through before you can get to that, point where you're creating a figure to put in your paper. So just to use Epic as an example, that's something that, Chris and I are familiar with as we use it at MGH. But Epic is this giant database, this giant electronic health record. It has three different sort of databases within it. It has Chronicles, Clarity, and Caboodle. Chronicles is the real-time database. So when you get a lab result, that was pushed into Chronicles from the lab, and that is real-time clinical information. Clarity is updated from Chronicles with some regularity. For a lot of places, it's every night. Chronicles essentially pushes to Clarity, and Clarity is a mirror of Chronicles and everything that was in it for the past day, or from with minus the last day. And then Caboodle is ultimately a sort of boiled down version of Clarity that's often used for administrative tasks. But essentially, if you want to query that, you have to use a coding language called SQL to essentially pull the relational databases out of Clarity and create something that is usable for you to understand. And so you go from SQL to the database, pull down your data, and then you want to use some statistical software, SPSS, R, Python, MATLAB, to essentially understand that data, apply statistical methods, apply machine learning AI, and ultimately get your result. And now we'll jump to our next topic, which will be talking a little bit more about omics, and I'll pass it back to Chris for that.
And so the first form of data that we're thinking of is our clinical data, right? And right now, a lot of that lives in Epic. But to make that even more powerful, we want to combine that with some sort of measure of the patient's acute physiology. And here we can really start to think about not only the biomarkers that we know about, our troponins, our lactates, etc., but discovering new biomarkers with omic technology. And many of you have heard this term and kind of used it probably, but may not have a deep understanding of it. Omics is a group of biological sciences that uses high throughput technologies to study the entirety of a specific type of molecule, or at least a plurality of a specific type of molecule within a biological system, right? Those may be genes, proteins, etc. We'll talk about that in a moment. But by looking at the entire system, or the systems level approach to research, we can get a more holistic view of all of those proteins or genes that are working at the same time, which one's active, how they're interacting. And with that, we can get new perspectives on research and diagnostics. The omics that we often talk about are genomics, right, which we're very familiar with ever since the Human Genome Project back in the 90s. And then you have transcriptomics, where you're now looking at RNA transcripts. And then as you approach the phenotype, you move into, say, proteins or proteomics. And then getting closer to the phenotype even still, you have metabolites and metabolomics. And of course, there are subsets like lipidomics and glycomics. And once you start having data from multiple sources like proteins and metabolites and genes, you can start looking at interactions between those in interactomics. And to this, I would also add other types of data. So people often talk about, for example, radiomics, right? So looking at radiologic biomarkers or imaging biomarkers and integrating that into these different types of omics. Most of these are now readily commercially available, although institutions like ours will also have resources to run those either in the hospital or at our research institutes. But you can go and essentially buy a service from one of the companies that offers proteomics, metabolomics, genomics, etc., send them your samples and get data back, often with a pretty thorough interpretation. They're not necessarily cheap, but they are available, and they're available to us for biomarker discovery in emergency medicine. We did this just recently, and those of you that attended the most recent SAEM meeting in Philadelphia saw Drew present this. We ran a proteomics analysis of acute pulmonary embolism with the goal of ultimately discovering new biomarkers. And this is just the technical aspects of how that's done, but essentially you have a targeted protein and you hybridize DNA that attaches to that protein, and then you can do PCR and PCR amplification in order to figure out which DNA hybrids are upregulated or downregulated, which then represent the proteins that are upregulated and downregulated in the system. And so we can now run over 5,400 proteins all at the same time and figure out not only the proteins that are up and down regulated in a patient with PE compared to someone who doesn't have PE, but the protein systems, in other words, the biological systems in which these proteins are all working together, which of those are upregulated. The same exact process works for metabolomics as well. And so this really provides us with a very powerful tool to make novel discoveries in our acute PE patients or other patients. We're going to put the data we get both from clinical sources and from either biomarkers or omics sources. And we're going to put that together. And because it's so rich and so detailed, to really truly analyze that well, we should leverage the power of AI. And now I'm going to turn you back to Drew, who's an expert in this.
Yeah. And so, at this point we have, biomarker data, we have clinical data, and we're looking to predict an outcome. We're looking to create precision diagnostics. And we don't really have the ability to go deeply into, AI and how it works in a short talk like this. This is things that, people spend careers doing. So my goal here is to give you a cursory understanding of what we're trying to do with AI and to give you some of the tools to think about doing it well and how you can gain those tools because, unfortunately, it is possible and in fact quite common for people to apply AI models without having a true understanding of what exactly they're doing with it. And so, just from a high level, artificial intelligence is a large umbrella. There are many things that fall into that umbrella. And essentially, we think about that as it sort of, moves down into things that are more and more detailed. And so one of the things that, I talk a lot about is machine learning. And machine learning, essentially what that is you're taking data and you're then asking it to predict an outcome. And the goal for that is to take a whole bunch of data and then be able to take a new piece of data that the model has never seen and predict an outcome. And that's what machine learning is. And there are sort of many sort of flavors of that, all the way down to these large language models that probably everyone has heard about. Open evidence in medicine and ChatGPT, Google is developing large language models, and so is sort of everybody else in the tech space. And so, but essentially, large language models are a form of machine learning and a form of artificial intelligence. And so I just wanted to give that sort of context to everybody. So, but the real challenge with AI is if you don't do it correctly and you don't think about what exactly you're doing, you may cause hallucination. And it's honestly more common than you might think that it is. And the reason that we do that and the reason that can happen is first off, insufficient data or bad data, which is why we talked about all of those downfalls with large data before. A faulty architecture. And what this means is, you know how to operate a statistical software, something like R, and you download a package for someone that's going to let you run a neural network. And then you just plug the data in and you churn it through and let their neural network do its thing, and you get the output. But for some reason, you didn't realize that there was a variable that needed to be set in the neural network because you don't really understand what it's doing, and you have a faulty architecture that gives you essentially the wrong output. Overfitting, this is something we think about in machine learning all the time. Overfitting is when we have too many inputs and we then get irrelevant outputs because essentially the model can fit a perfect function to every single variable that you put in it. And so you get a model that essentially only tracks the data that it was trained on, and you get really, irrelevant outputs at the end. And finally, generation methods, and this really is where the large language models come in because they tend to prioritize fluency over accuracy. And so you get a beautiful sort of text output, but it can kind of be garbage because you lose some of the accurate information in there. And so finally, you get the chain of AI hallucination. So essentially, you have data problems, you have model problems that result in a hallucinated output, and ultimately a failure of your AI model. So, how do we fix this? Well, it all starts with ethical standards for AI in healthcare. And fortunately, the World Health Organization set 6 ethical standards for AI in healthcare. And anytime that someone runs an AI model on healthcare data, they should be thinking about all of these things. And so, first and foremost, we want to protect autonomy. We want to protect our patients. We want to protect their data. And we want to think about that and how we can make sure that, no one can get identified from any of these things. We want to promote human well-being, human safety, and the public interest. Our goal with using any healthcare data should be that we're trying to promote public health, promote public safety. We want to ensure transparency, explainability, and intelligibility. And this is a really big one. A lot of AI, things like LLMs, things like neural networks, you can't really get under the hood, so to speak. These things make decisions, they develop associations between data and, parts of the model that we can't understand. And so thinking about how we can really create machine learning models that we can go back and actually look and see what they're doing is actually incredibly important. We want to foster responsibility and accountability. If you're, creating an AI model, you really should feel responsible for the outcome and where that model goes and making sure that it does all of the things that you say it's doing and that it's been well tested on many different types of data to make sure that it works. Ensure inclusivity and equity. So that comes back to our data sets, making sure that we have, data sets that are unbiased as much as possible. And we want to promote AI that is responsive and sustainable. All right. So what does that come down to? So it comes down to AI education. And so if you are someone that is trying to, build a career in emergency medicine and innovation with a component of AI and predictive modeling, you really want to get good education. There are a lot of online resources for getting good education, and I don't want to, endorse anything in particular, but I've put a few free online softwares up here that are really pretty good. One of the reasons that they're good is that they partner with universities that have, academics that do machine learning, provide some of the teaching in those courses. And so these are people that have dedicated their career to AI and machine learning. And they're offering that to the public to help inform and educate them. You can also consider formalized training, such as a master's or a PhD in computer science or engineering, a fellowship in informatics, research, or innovation. And finally, if you're someone that, say, wants to use this and sort of sees the value of it, but you're in a spot in your career or in your life where you don't have the time to dedicate a tremendous amount of time to learning how all of these things work, that's where university and industry collaborators come in. There are people that do this for a living and they're always looking for, the next great data set that we can use to push healthcare forward. And this is a great time to find those collaborators and talk about how you can bring your emergency medicine expertise to them and they can bring their AI expertise to you, and it's fruitful for everybody all around. So finally, for AI and emergency medicine, we have the fantastic opportunity coming up at our next conference in May 2026, where we will have a consensus conference on AI and the future of emergency medicine. And for anyone that's interested in this space, this could be something fantastic to attend. The consensus conference, the agenda has not been set, but we'll likely be talking about where we see AI in the field of emergency medicine in the next 10 or even 20 years. So if this is something that's interested to you, it's a great opportunity to meet a lot of the players in AI and emergency medicine. Finally, I'll pass it back to Chris and he'll talk about how you can tie all these things together. So what is our vision for an EM career in clinical innovation and research translation?
Yeah, so this is one of the reasons I think that this particular webinar is kind of fun for us because we spend a lot of our time in the weeds and talking about the details, but this is an opportunity to talk about kind of our big vision and where we think emergency medicine research and science and innovation may be going and how we're going to help take us there, hopefully in the future. So this is where we think clinical innovation and research translation can go in emergency medicine and what we're working towards. So if you're thinking, you know what, this sounds great. I want a career that will allow me to innovate, translate from basic science to clinic to the bedside. I want to be part of this research that really guides who we are as a specialty moving forward. Then you're going to need a few things, right? You're going to need access to data on the upper right. And that data is the stuff we talked about already, the clinical data, the biorepository data. But there's something we didn't really mention, which I think is really important, and that's translational data that gets us from physiologic or animal models back to humans. And that's important for reasons that you may not think. In the PE world, for example, Patients come in with their PE. Very few patients have their PE while they're in the ED, so we will never know what their baseline status was. Animal models, physiologic models, in silico models will enable us to actually look at the pre-disease state and ensure that the things that we're finding in our clinical research are actually part of the causal pathway rather than maybe being less reproducible predictors. You're going to need some computational resources in order to do this work. Fortunately, most of us with our university settings have access to that. And as Drew mentioned, a lot of this is becoming easier over time, but this is still going to require a bit of computing power. And then I think the most important thing to recognize is that this is not likely to be something that you can do on your own. You will have some expertise, and you will certainly have expertise in emergency medicine, which I do not think that we can downplay at all, because most of the other people that you're going to work with in this field are people who are experts in artificial intelligence or basic science biology or high throughput omics or data analytics and statistics. They're not at the bedside, right? If we're studying PE or we're studying intracranial hemorrhage, most of those people have never seen a patient with a PE or an intracranial hemorrhage. So I want to say this because I have felt these doubts myself in my career and wondering why I felt like the stupidest person in the room. And it took me a long time to remember that no, I bring something to the table that the people that I work with don't. But you should work with those people because they bring amazing expertise. And if you create collaborations with your AI experts and your data scientists and your basic science researchers, you can create a team of diverse skills and talent that will really drive this science forward. And so that's what we're trying to do as we look forward for the next 10 years at the Center for Vascular Emergencies at Mass General. Again, we've been focused on the care of DVT and PE patients, but we are expanding to other things, and we are creating those kind of collaborations and creating those kind of resources to do this work as we move forward. The clinical data resource that we're focused on at the moment is something called Archive. And here we have to give a huge shout out to our friends and collaborators at Vanderbilt University, Bo Stubblefield and Jesse Wren. Together, we are building this resource, which is an automated registry of patients who are evaluated for possible cardiovascular disease, and in particular PE and DVT. And we're now rolling that out at 11, 12, 13 centers throughout the United States. And very soon we're going to have one of the largest, most robust databases of patients tested for cardiovascular emergencies in the world. And it's super exciting. At the CVE, we are also highly invested in our own biorepositories. So we have been collecting blood samples on patients who are tested for possible PE and patients now who are tested for abdominal diagnoses, and we're moving on to other things as well. So I think these are going to be incredibly useful as we move forward. Our approach is to focus on particular phenotypes, PE, for example, and then enroll patients who we think make excellent controls for the patients with the disease. So in particular, we're enrolling patients who come to the ED, have enough signs, symptoms, and risk factors that the doctor thinks they need a workup for PE, does the actual test, and then our control patients are the ones that just happen to have a negative workup, right? So that's a very good control when you want to hold that up next to someone that does have a PE to help you really differentiate the patients with the phenotype from the patients that don't. And so that's our approach. It's not the only approach to biorepositories and biobanking, but it's important that we have that resource, as many other people do. Informatics and computational resources, again, I'm not going to go into the weeds in this. You do need to work with your informatics colleagues to get access to things like Epic and the data that are in there, and you have to have the computational resources to analyze those data. And then this is Mark. He's a colleague of mine and a mentee of mine who's in Denmark. And they happen to have a really fascinating animal model of PE where they coagulate blood in rubber tubes and then inject them back into an animal model to create a large PE, which we can then study using a lot of the same methods we talked about before, proteomics, metabolomics, biomarker measurements, etc. And now we can know which of those things we're seeing in people are actually changing from the pre-disease state to the post-disease state, which you can almost never know when you're doing clinical research in the emergency department and patients show up because they have the problem when they walk in the door. So this is another resource that we've developed at our CVE and I think is worth other people thinking about. And then lastly, probably the most important thing are our friends, right? That's probably a good lesson in life. Your friends are your friends and your family are your most important thing. And we have our work friends and work family. So we have colleagues at the Broad Institute, which is a large multi-center institute that focuses on omics. So genomics, proteomics, metabolomics primarily. We have our friends in the Department of Computer Scientists and the Data Action Lab at Harvard Computer Science, who are experts in machine learning. We have similar experts in machine learning and, in fact, sort of the economic impacts of some of these things that we're working on at MIT. And then we have our basic science and translational friends at Aarhus. And this is our network that we're building. And I'm sure that a lot of you who are listening think that you probably can go across the street and create a similar network to ours. And I would encourage you to do that because I think the more folks that we bring to the table, the faster we're going to be able to improve the diagnosis and treatment of undifferentiated ill patients in the ED. Again, the last thing I'll mention is that we think this is the future. And if this is the future, then we're going to want to train people who know how to do science and emergency medicine for the next 20, 30, 40 years. I'm going to be around for some part of that, but probably not the whole thing. And so I hope that we can provide these resources and our contacts and our expertise to other folks that want to learn. And so we've created a fellowship at MGB, and we'd love to hear from you as we move forward. There's a QR code if you're interested in getting in touch with us. That'll lead you to a connect page on our website. You can always get us through our email. And I just want to thank everybody for your attention. I hope this was, mildly interesting. And please reach out if you'd like to learn more.
Awesome. Thank you so much, both to Dr. Cabral and Dr. Bierenkott. I will open it up first and see if anybody has any questions that's out in the audience. And if not, I have a few, so we'll see. I'll just go ahead and start us off. I feel like might as well get everyone warmed up. One thing that came to mind that was really interesting as you were mentioning, the various contributors to making an accurate model, and one of those being data quality and sort of these large data sets. Without getting overly political, I think there's been some concern sort of around the medical and the scientific community that some of these, and even beyond that some of these national organizations that have previously overseen these large data sets may not be being managed either efficiently or to the best of their ability. Do you have any concern that's going to impact any of the data sets that you use for your research or have negative effects down the line for the kind of work that you're doing?
So for the work that we're doing, I think that it will, likely be unaffected. We are very fortunate in that the data that we're using is what I would call homegrown. And so we are extracting the data ourselves and have that data set and that database, which is primarily the data that we're using. I mean, that said, in the event that, data sets that are being collected are not, representative data sets of whatever you're trying to predict, there will be issues down the line with that. And that's exactly the concern with big data.
Yeah, and I'll follow up on that to say that as we are home growing our database, we are taking great care to make sure that we validate the key variables that we are looking at. And so, for example, have we've created algorithms that essentially read the reports of radiology scans and provide those results to determine who has a disease and who doesn't. We validate those every time we roll this out to a new site, and we do it with human adjudication to make sure that our models are working. And I think that's just what we do. But I think even if you use a data set that you've downloaded from another source, it is important that you do some sort of validation to make sure that passes the sniff test, right? And the genetic research that I do, we always run our associations to make sure that we find things we expect to find, right? If we ever have a database that, maybe we're doing epidemiology, we want to know if the data look generally good, we'll run to see if BMI is associated with PE because we know it is. And if we don't see that, then there's a problem, right? And so no matter what data set you use, I think it's important that you apply some quality control and make sure that your data passes that sniff test, or else you're going to have the same garbage in, garbage out problem that Drew talked about.
Awesome. Thank you. I guess another one to throw out there for anyone who's listening either in real time or recorded, who's maybe a student or a newer trainee and feeling very inspired by what they heard and thinking this is something that I could see myself getting into. And you really did an excellent job of laying out a pathway to get there. But maybe if you're not quite at the step where you're doing a fellowship in computer science or whatnot, what would be your advice as to kind of first steps you could take to get your foot in the door of this kind of work?
I mean, the first step is to find the people that are doing it. And this is honestly one of the reasons that we're here giving a, RAMS SAM webinar is, we are, interested in talking to the folks that are interested in doing this, but at the same time, SAM is full of people who are doing this sort of work. And so, come to SAM and meet those people and make those connections, because the way to get your foot in the door with this is to find the people that are doing it and to find that mentorship and the people that can sort of, advise you on what you should be focusing on. Because, we showed that whole diagram of the expertise and collaboration that you need. There is no way that you can have all of those things. And so figuring out which one of those pieces of expertise you are going to have and who you're going to collaborate with for the rest, I mean, that's big sort of career, advice stuff. And it's something you need a mentor for to guide you through.
Yeah, odds are you need multiple mentors. And I've said that my entire career is that you almost always need multiple mentors. And if you're still unsure because you're new to the field and new to medicine and new to the specialty, talk to someone who's senior at your place. Odds are they know people, right? They may not be doing the thing that you're doing, but they will know who's out there, and because they've been around a long time like me, or reach out to us. And even if you don't wind up working with us, we'll put you in touch with people that are near to your home institution or near to your personal interests. And I don't know why everybody wouldn't be interested in PE, but maybe somebody is interested in something else, right? And but we know who's doing that work, and I'm sure that the senior people at your institution do as well. And so I completely agree with Drew's comment. It's just talk to folks and find out who's doing it, knock on their door, and be persistent.
Awesome. Well, I think that's really excellent research and just life advice in general. So, two for the price of one. We appreciate that. To wrap it up, as you said, your focus has been PE and vascular emergencies, and there's been some allusions to something else. So, I'm curious if you're willing to give any spoilers. Are there any sort of something else's in the pipeline that you think either you guys are working on or maybe would be a ripe area for somebody else to try and jump in on the ground floor of?
Yeah, it's a great question. And it's not a spoiler. We're open to a lot of different things. The really cool thing about this process is that with the ability to generate automated data and the ability to biobank from multiple different types of patients, this has sort of a rinse and repeat quality to it, right? You can take our VTE phenotype out and replace it with a different phenotype. So one of the things that we're interested in that I mentioned is abdominal diagnoses, right? It's one of the most common things that present to the ED. We personally find that starting out, picking a diagnosis that's relatively discrete is easier, right? So syndromic things like TIAs are challenging, right? Because ultimately you have to have some ground truth diagnosis to say this is a PE and this isn't. And PE is nice because generally speaking, it's based on the CAT scan result. And so that makes our lives a little bit easier. And so if you're thinking about, well, I'm really interested in, psychiatric illness, for example, you may find yourself challenged because knowing what that ground truth is a little hard, and you may ask yourself, okay, well, maybe within that, I can find something where I know I can say this is the disease and this isn't. This is the phenotype and this isn't. Because that, I think, ultimately will make your life a little bit easier as you move forward with this.
Awesome. Yeah, I think that's excellent advice. And I want to thank both of you again for coming out and doing this. And huge thanks as well to the SAM Research Committee for sponsoring another fabulous research learning series. I think if we don't have any other questions, we can probably wrap it up for the evening.
Yeah, it's been super fun for us. And like I said, it's a great chance for us to talk about some big picture vision. And it's really been a pleasure to share it with y'all.
Awesome. Thank you so much.
Thanks very much.
Thank you all so much. This was awesome.